We've looked at carbohydrates, we've looked at lipids/fats. Now we'll turn our attention to proteins. Proteins contain the elements carbon, hydrogen, oxygen, and nitrogen, so CHON, C-H-O-N, and some proteins contain sulfur. Now, the basic building block of a protein, its monomer, is known as an amino acid, and there are 20 different amino acids. The reasons why 20 different amino acids exist is solely down to their structure, which consists of a central carbon atom joined to a hydrogen atom, a carboxyl group, which is COOH at one end, and an amine group, which is NH2 on the other end. There's an R group, and that just means that this is a variable group that sometimes contains sulfur, sometimes contains different combinations of elements, and this is what actually gives you the variety in your amino acids. Now, amino acids joining together forms a polypeptide, and the way in which they join together is through a condensation reaction, which means there's a loss of a water molecule, H2O. So where is that water molecule lost from? Well, we lose an OH from one amino acid's carboxyl group, and we lose an H from the neighbouring amino acid's amine group, and together they join to form what's known as a peptide bond. So you'll see that peptide bond existing between the carbon of one amino acid and the nitrogen of another. If you have two amino acids joining together, we have a dipeptide, three amino acids, tripeptide, many amino acids, a polypeptide. Now, let's link the structure of an amino acid with the end structure of a protein. And a protein has a primary, a secondary, a tertiary, and a quaternary structure. So what is the primary structure of a protein? It's simply the sequence of amino acids. What is the secondary structure? Well, it's when that chain of amino acids known as our polypeptide folds together in order to form an alpha helix or a beta pleated sheet. Now, the shape of that alpha helix and that beta pleated sheet is held in position through hydrogen bonds which occur between amine and carboxyl groups on neighbouring amino acids. The tertiary structure is the 3D folding of our polypeptide chain. This is held in position by ionic interactions, disulfide bridges, that means between two sulfur atoms, hydrophobic and hydrophilic interactions, and more hydrogen bonds. So clearly, the constituent elements of our R group on our amino acids are very heavily going to dictate the final shape of our protein because they'll dictate what type of interactions hold the polypeptide chains together. The quaternary structure simply means that two or more polypeptide chains are folded together to form a final protein, which can function. So remember, an enzyme, for example, is a protein, and therefore the active site will be brought about through the tertiary and quaternary stages of the folding of a protein. So I've already mentioned enzymes. Now, enzymes are examples of what we call globular proteins. A good example is the enzyme ribisco, which is used in photosynthesis. Hormones are examples of globular proteins. For example, the hormone insulin, which is responsible for lowering blood glucose levels by converting glucose into insoluble glycogen. Another good example of a hormone is glucagon, which increases blood glucose levels by converting glycogen into glucose. Let's look more closely at the structure of a globular protein. So remember, I said that enzymes and hormones are both examples of globular proteins. So, as I've said, a protein structure is made up of its primary structure, which is simply its sequence of amino acids. The secondary structure is when it folds either into an alpha helix or a beta pleated sheet, and this position is held through hydrogen bonds. Then in the tertiary structure, this is when we get our 3D shape of our polypeptide chain. Depending on the various R groups, this shape could be held in position through ionic bonds, hydrogen bonds, disulfide bridges, or hydrophobic interactions. And then when two or more polypeptides are linked together, we have what's known as a quaternary structure. And here we have the addition of a prosthetic group, which simply means the addition of something which isn't a polypeptide, so doesn't consist of amino acids. There are structural proteins, such as the fibrous proteins, keratin found in our hair, and collagen found in our skin. If we're being asked to distinguish between fibrous and globular proteins, so fibrous ones including the keratin in your hair, collagen in your skin, are globular proteins such as hemoglobin. The first thing is their shape. Fibrous proteins form long chains which are linear. Globular, as the name suggests, are more spherical. Fibrous proteins are usually insoluble. Globular ones are usually soluble. Fibrous ones provide strength. Globular ones may form hydrogen bonds with water, which increases their solubility. Fibrous proteins have structural roles in your hair and your skin. Globular proteins may be involved in catalysis if they're enzymes or transport roles, such as hemoglobin used to transport oxygen. So looking more closely at the fibrous protein collagen, we know that peptide bonds form between amino acids. That's standard. Now, every third amino acid is the same, which is glycine in collagen. So glycine is the name of one of the 20 amino acids. It forms what's known as a left-handed helix. The glycine presence means that the polypeptide chains pack close together to form fibrils. Hydrogen bonds occur between 3 polypeptide chains, and there are a few hydrophilic R groups on the outside of the molecule. In terms of the structure of that hemoglobin used to transport oxygen in red blood cells, your primary structure remains as you'd expect. The secondary structure consists of an alpha helix and small regions of beta-pleated sheets held together by hydrogen bonds. In the tertiary structure, polypeptide chains undergo further folding through a variety of interactions. These polypeptide chains are held in a 3D shape. There are hydrophilic, water-loving R groups on the outside of the molecule of hemoglobin, whereas hydrophobic, water-hating R groups exist on the inside. And then lastly, the quaternary structure consists of 4 polypeptide chains with 1 heme group. And that contains our prosthetic group, so something which isn't a polypeptide, which in this case is Fe2+. Fe2+ refers to heme, is part of the reason why it's so important that we have plenty of iron in our diets, because that contributes to us having blood which can transport lots of oxygen on the red blood cells. The fact that some amino acids are polar and non-polar conveys very different properties in the various proteins they're used to make. And it's the variable R group which will affect the polarity of the amino acid in question. Now, polar amino acids on the outside of proteins make them soluble in water. Polar amino acids in channels and membranes allow them to transport polar substances across the membrane. And the polarity and the non-polarity on surface amino acids on proteins determines their interactions with other molecules such as substrates, hormones, and signaling molecules. Now, you've met the word denature previously. We'll just add a little bit of extra detail here. So what does denaturation mean? It means a deformation of the 3D shape of a protein, so in effect an alteration in its tertiary structure. If the protein in question is an enzyme, then that denaturation will obviously affect the enzyme's active site, deforming it, meaning that the substrate molecules can no longer fit. What causes denaturation? High temperatures and a rapid change in pH. Notice that high temperatures usually cause an irreversible denaturation, one that cannot be reversed, whereas changes in pH cause denaturation, which is often reversible. And why does that high temperature have such an irreversible impact on that protein structure? Well, it's because the high temperature breaks intermolecular forces, damaging the 3D structure by breaking things like disulfide bridges and hydrogen bonds. How are we going to test for the presence of protein using the bioarray test? Now, bioarray reagent is actually made up of 10% sodium hydroxide solution and 1% copper sulfate solution. So when you add the bioarray reagent to the sample, if it turns lilac, that means you have protein present. Now, how can we identify various amino acids in a sample? Well, we can use thin layer chromatography. This is something you'll meet in A2 chemistry if you choose to study it. So how this works is you add your amino acids to one end of a layer of silica gel. This is known as the stationary phase. You then submerge the end in an organic solvent. That organic solvent starts to soak up the stationary phase, up the silica gel, and with it, it will separate those amino acids into their various components. Due to differences in their polarity and their solubility, they'll travel at different speeds. And by calculating an RF value, which is the distance travelled by your component divided by the distance travelled by your solvent, you'll be able to use that number calculated for your RF value once it's been compared to a reference table to identify a particular amino acid in your sample. The diagram shows the primary structure of part of a polypeptide. Each shape represents an amino acid. Identical amino acids have the same shape. Name the type of peptidase which will hydrolyze the bond labeled G in the diagram above. So we're hydrolyzing the bond between 2 amino acids. It's an endopeptidase. Give the number of different R groups in the polypeptide shown in the diagram above. So really that means how many different amino acids do we have? 1, 2, 3, 4, 5. That's one amino acid type. That's the second amino acid type. Here's a third. I've crossed them out to make sure I've got them all. It's 3. The scientists used an enzyme to digest a polypeptide containing 101 amino acids. The digestion produced a range of smaller polypeptides. The scientists determined the number of amino acids in each of the polypeptides produced. He also counted the number of polypeptides of each length. The table below shows some results. Use the information in the table above to calculate the number of polypeptides, 6 amino acids in length and 20 amino acids in length. So just work out how many amino acids have been used up. That's 10 here. That's 45 here because I've multiplied these two numbers together. So added up, that is 55. So do 101 minus 55 to get 46. And then look at the numbers we have left over, 20 and 6. So how do I get 46 out of that? Well, I think I'm going to do 2 lots of 20 and 1 lot of 6. And then in total, that should equal 101. Describe how the structure of a protein depends on the amino acid it contains. That's worth 5 marks. So we're going to talk in great detail here about primary, secondary and tertiary and quaternary structures. And we're going to talk about how the structure of a particular protein depends on the relative position of the R groups of amino acids, because that will then create the kind of interactions such as hydrogen bonds and disulfide bridges between various amino acids. So the structure of the protein depends upon the position of R groups in amino acids. Let's go into the various types of structure. The primary structure of a protein is the order of amino acids. The secondary structure is the folding into alpha helices and beta pleated sheets, which are brought about by hydrogen bonds between amino acid chains. The tertiary structure, that means tertiary, is formed by interactions, e.g. disulfide bridges between R groups of amino acids. Active sites in enzymes may be produced here. So this whole time we're linking back to how the structure of the overall M protein depends very much on the amino acids it contains, because it's their R groups which will dictate the type of interactions between the amino acid chains. Figure 2 is a diagram of one SGLT1 carrier protein. Figure 2 shows the SGLT1 polypeptide with the amine group at one end and the carboxyl group at the other. Describe how amino acids join to form a polypeptide. So there's always one NH2 at one end and a COOH at the other end. You may use a diagram in your answer. And I'm not one for diagrams, but I think I will just do this. So we know that the amino acid contains central carbon, it has an R group which is variable. One end is the carboxyl end, one end is the amine end. I draw another amino acid next door. It's a condensation reaction, which means that there's a loss of a water molecule. So I'll show you how that occurs. We lose the OH of the carboxyl end, the H of the neighbouring amino acid's amine end, and then we stick together these ends. So just to show that a bit more neatly, let's just move this amino acid along, and this is what we call our peptide linkage or peptide bond.